So we have talked about DNA a few times throughout this year. We know it is an organic compound. We know its basic functions, and we are also familiar with its twisted shape. But now we need to look more detail about its structure. That's important because the structure allows it to do its job. So DNA stands for deoxyribonucleic acid. Quite a long name, which is why we do abbreviated DNA. So there is the D for deoxyribo and the NA for nucleic acid. This is actually really descriptive of the components of this molecule that we will see. Notice deoxyribo, that's part of the name for deoxyribose, which is the sugar, and the type of organic compound, the nucleic acid. So DNA has a couple of important functions. It is the instructions for making proteins. So your DNA has those instructions to tell your cell how to produce the many proteins that you need. It is the code for your traits or the blueprint for life. So it gives you your characteristics, your hair color, your eye color, your skin tone, because it does make those proteins, or it has the code for making those proteins. So it is the molecule of heredity or inheritance. So it's passed from parents to offspring and generation to generation to carry those traits down. As we said, it is a type of organic compound. It is an example of a nucleic acid. So you might remember we talked about 4 different types of organic compounds this year: carbohydrates, lipids, proteins, and nucleic acids. This is one of our examples of a nucleic acid. So DNA's structure is a double helix. So it's double in that it has 2 strands, and helix means it is twisted. So we're pretty familiar with this basic shape of that double-stranded molecule that's twisted, and it resembles a twisted ladder or maybe even a spiral staircase. Those 2 strands run parallel to one another, which means they run side by side, and actually they are anti-parallel. So one runs opposite of the other. There are rungs that connect the 2 parallel strands, so there are specific parts of the DNA structure. We'll talk about the building blocks and the parts of those building blocks, but the rungs connect those 2 strands to make that ladder structure. So we can see the 2 strands here in blue. They are connected, one from the left to the right in this case, by these rungs, if we look at this as a ladder. And notice the 2 strands are twisted as we move down this molecule. So let's take a little bit of a closer look at the building blocks of the DNA structure. DNA is made up of nucleotides. That is the monomer or building block of our nucleic acid. We talked about amino acids being the building block of proteins, monosaccharides being the building blocks of carbohydrates. The nucleotide is the building block of a nucleic acid. So each strand of DNA is made up of a series of nucleotides bonded together. Nucleotides have 3 components, and if you look to the picture to the right, you can see these 3 components. One is a phosphate group. That is a phosphate group in the circle here. A 5-carbon sugar. The name of that sugar is deoxyribose. You can see that down here at the bottom, and that also is part of its name. And then a nitrogenous base, which means nitrogen-containing base. You might remember earlier in the year when we talked about the elements that make up our organic compounds, how DNA has carbon and hydrogen and oxygen, but also nitrogen and phosphorus. So there is the phosphorus and there is the nitrogen, and that nitrogen is found in the nitrogen-containing base. The bases actually come in a couple different shapes, but the phosphate and the sugar are consistent, the same for every nucleotide in DNA molecule. So you should have a picture in your notes of a nucleotide. So label that it is a nucleotide, but also its parts, the phosphate, the sugar, and the base. So nucleotides come in 4 different forms because there are 4 different bases. So the only difference from one nucleotide to the next would be the base that it contains. Our bases are adenine, which is abbreviated with a capital letter A, thymine, capital letter T, guanine, capital letter G, and cytosine, capital letter C. So each nucleotide has one of 4 bases, only one each. Notice that the adenine and guanine are purines, where thymine and cytosine are pyrimidines, and that deals with their structure. So you can see adenine and guanine have 2 rings that make up that base, where thymine and cytosine have only 1. And we're going to see a consistent shape because of these combinations of the bases. So specific bases pair all the time. There's something called Chargaff's rule, which Chargaff was a scientist, and he studied the amount of each of these bases in a whole bunch of DNA strands. So he took samples from different organisms and figured out the amount of cytosine, the amount of guanine, the amount of thymine and adenine in each of those DNA sample molecules. And he noticed that every time he found cytosine in a specific percentage, that molecule also had about the same percentage of guanine. Same thing with thymine and adenine. So that led him to Chargaff's rules, which state that cytosine and guanine are found in equal amounts and therefore must be paired together. Thymine and adenine are found in equal amounts and therefore must be paired together. So there's our base pairing rules, that adenine always pairs with thymine and guanine always pairs with cytosine. So we always have a double ring base paired with a single ring base that helps keep that DNA molecule consistent in shape. So a strand can actually just be represented by the series of bases or those letters, because the sugars and the phosphates on the back side of the DNA are always consistently the same. So the only thing that's different really is the DNA bases in the middle, and that's what actually gives the code for our traits. So if we saw a strand ATT, ACA, and so on, its complement you can figure out based on its base pairing rules. So if there is an A, across from that A will be a T. So notice that the complements here make up the other strand, or the matching anti-parallel strand, and we can represent that by simply a series of the bases, or those letters that represent those bases. So nucleotides form a string when they are connected to each other. So if we look at this phosphate, sugar, and base, that's one nucleotide connected to the next nucleotide. So nucleotides link to one another by a very strong bond. You might remember covalent bonds. So we want the covalent bonds connecting them because we want this to be a strong bond to hold the nucleotides together, forming that single strand. But between the strands, we have weak hydrogen bonds. So it might sound strange that we have such a weak bond in such an important molecule. However, we do want to be able to separate this sometimes to read the molecule. So in order to read the molecule, we have to unzip that molecule or break the bonds between so we can actually get to this series of bases. So weak hydrogen bonds are found between the bases, and that helps to connect one strand to the other strand, making that double-stranded molecule. Okay, so we have our DNA structure here, and you have a picture of this in your notes. So we'll take a few minutes to see if we can label the structure to practice identifying the parts. So we'll label the deoxyribose sugar, the phosphate group, a base. We'll circle and label a nucleotide, covalent bond, and hydrogen bonds. So pause the video now and see if you can try this, and then come back and we will see if we can check our work. Okay, hopefully you have tried this, so let's check our work. Right here we have a deoxyribose sugar, so the sugars are all the way down the outside every other molecule. Notice the structure, it's this blue structure here with a 5-sided structure. It is a 5-sided molecule because it is a 5-carbon sugar. Alternating on the outside, we have our phosphate groups. So here it is in yellow. This circle here is our phosphate group, also alternating all the way down the outside. That makes up the sides of our ladder. The bases are in the middle. So here is one base, here is another base. Notice, remember, some of our bases are single rings and some are double rings. So here is a base and here is a base. A nucleotide is made up of 3 parts, so it's going to be a phosphate group plus a sugar plus a base, just one of the bases. So adenine plus sugar plus phosphate. Covalent bonds are going to be any of these solid lines. So right here is a covalent bond, right here is a covalent bond. Here's a good example because it shows you the connection between this nucleotide and this nucleotide, connecting between the phosphate and the sugar. And then hydrogen bonds are these dotted lines in the middle, connecting the bases and therefore connecting the 2 strands to each other. All right, here's another practice, but this ladder, notice, is just turned on its side. So if this is our 5-sided structure, that is our deoxyribose sugar. Along the outside here, alternating with the sugar, we have our phosphate groups. Remember, cytosine pairs with guanine, so if this is cytosine, that makes this double ring base here guanine. If this is thymine, remember thymine pairs with adenine, so if this is thymine, it pairs with this double ring base, adenine. This DNA molecule is simply on its side, so right here is one strand, here is the other strand, and in between are the bases or the rungs. These solid lines here are covalent bonds holding a nucleotide to the next nucleotide, and in between these dotted lines here are our weak hydrogen bonds found between the bases, holding the bases together and therefore the strands together. Weak so that we can break them to read the molecule.